Thank you, Laura. Thank you, everyone, for joining in. It's a real pleasure today to introduce Luca Kuziel, who worked at the ranch for close to 7 months. He started early in 2022 as a sugarcane intern and switched to work on his project on West Indian Marsh Grass and the impact of control method on soil and water properties. A few words about Luca. Luca came to us from Princeton University. He majored in ecology and evolutionary biology with a minor in environmental studies. He actually worked previously as an intern in Mozambique. So that was a pretty big change for him to come to Florida. And he's now currently working in New Mexico at the probably not going to butcher it, Carlsbad Caverns National Park with the Park Services. So he's been there for maybe a couple of months already. We were sorry we couldn't get to do that internship prior to that, but Luca was a bit unlucky and ended up finishing his project right when Ian started here. So we're very happy to see Luca again. And we hope that he's having a good time in New Mexico. His goals are to pursue a career in conservation and restoration ecology. So without further ado, Luca.
Thank you, Greg, for that lovely introduction. Yeah, this is almost 2 months to the day after I was supposed to give this presentation, but that's fine. Here I am. So yeah, hi, everyone. My project is on the impacts on nutrients of West Indian marsh grass management methods. So West Indian marsh grass, or Hymenacne amplexicaulis, is a grass native to South America and the West Indies. It's good forage for cattle. It's highly palatable, digestible, and nutritious, so it can be beneficial for ranching. However, it's also a Category 1 invasive species, which according to the Florida Invasive Species Council, is a species that displaces native species, changes community structure or ecological functions, or hybridizes with natives. In this case, West Indian marsh grass displaces a lot of native grasses. It removes resources for and it blocks water from entering waterways, and it blocks waterways as well physically. So different West Indian marsh grass management strategies typically include herbicide like glyphosate or imazapyr. It can also include removal by machinery or flooding, drowning, or shading it, which is difficult on a landscape scale. It can also, there's a potential for biological controls by cattle, which I will talk about. West Indian marsh grass at Buck Island Ranch. It's on the ranch. There was a study done on dispersion and where it's located by Bouchard et al. So you can see in this map the improved pastures, which are pastures that were fertilized, are the brown ones in the middle, and then the semi-native, which weren't as fertilized, are the green on the edges. And then the blobs in the map are the 600 wetlands on the ranch, and the green wetlands are ones where West Indian marsh grass was found, and the orange wetlands are wetlands without it. There's also been work done on the ranch on seed banks and seed bank persistence by Akeem. And then there is also a National Resource Conservation Service exotic control project on the efficacy of different methods of West Indian marsh grass management that's led by Greg, that Akeem also works on. So that control project, as I said, examines the efficacy and cost of different management methods. And there are 10 experimental plots set up in wetlands, 4 at Buck Island Ranch, 3 at Santa Rosa Ranch, and 3 at Archbold's Reserve. And each plot has 4 subplots with the different treatments. So one treatment is just leave it alone, let it grow. Another is let cattle graze it. There's also a combination of grazing and applying herbicide. And then the final treatment of not grazing, but applying herbicide. And this just looks at, does it kill the plant and what's the monetary cost? But of course, it could also have other impacts on the environment. So nutrients in the environment are very important. Fertilization of rangelands and farmlands is pretty critical for agriculture. Big nutrients that are applied are phosphorus and nitrogen. And Buck Island Ranch fertilized its improved pastures from the 60s to the 80s with phosphorus. Nitrogen is still applied every few years if the price is right. And while those nutrients increase plant productivity, it can also have negative impacts on the environment, such as eutrophication of Lake Okeechobee and the Everglades. And eutrophication is where a large influx of nutrients, so often runoff from a bunch of different ranches, bringing nutrients into one water body, increases the productivity of algae, and then you're left with algal blooms. And once all that algae starts to die, its microbes start to decompose it. Those microbes use up all the oxygen in the water. You have less oxygen, and then all the animals die. So it's not great. And there are different projects to try to address this in Florida, such as the Northern Everglades Payment for Environmental Services, or NEPS program, which pays ranchers to hold water back on their properties to prevent the nutrients from getting to Lake Okeechobee and the Everglades. There have also been studies done on the ranch on nutrient uptake abilities by various plant species, such as the Grove Project, which is using a former orange grove and watering it with water that's very high in nutrients, and using that to water plants that are then harvested and given to the cattle supplementally in winter in Florida. And so there was a study done to see which plants would be best at removing the most nutrients from that environment once it was watered with the high nutrient water. So from all of this, I saw a few potentials for research. West Indian marsh grass is highly invasive, so how do we control it? It's also highly nutritious, but those nutrients have to come from somewhere. So how does West Indian marsh grass impact nutrients in the environment? And it's also highly controversial. So how do we ensure that everyone's needs are met? So my research used the existing study on control methods to measure impacts on nutrients in the environment. And hopefully this can help inform the NRCS study about impacts on nutrients that could help decide on a management plan and provide scientific evidence about the ability of West Indian marsh grass to remove nutrients from the environment. So my research questions: one, how do different methods of controlling West Indian marsh grass impact soil and water nutrients? And two, how does West Indian marsh grass compare to a native equivalent? So on the left is West Indian marsh grass, and on the right is maiden cane, or Pancum hematomen, which is a native wetland grass that also, similarly to West Indian marsh grass, forms dense monospecific stands. They play a similar role in the environment, and maiden cane is displaced by West Indian marsh grass. So it seemed like a good species for comparison in this study. So my design overview: to answer my first question about the impact of treatments on soil and water, I created an experimental mesocosm, applying the treatments that were in the field to buckets of the plants. And that allows me to more clearly tease apart different impacts and sort of control all of the other factors that could impact it out in the real world. And that also helps me answer the second question, comparing the two different species. So I planted the two different species in buckets, and I applied the treatments from the field, except instead of letting the cattle into the shade house to eat all the plants, I used scissors to clip them to simulate grazing. So I had clipped and unclipped of both maiden cane and West Indian marsh grass. And then for the West Indian marsh grass, I also had herbicide and clipped and herbicide. And I didn't herbicide the maiden cane because it's a native species. There's not really a real world situation in which it would be herbicided. And then I also had a few buckets that just had soil and water, no plants. And then I had a bucket with just water. And from those, I collected water samples and measured nutrients from them. I collected soil samples and measured nutrients from them. And I'll tell you what those nutrients are in a moment. And then from the plants that I clipped to simulate grazing, I also measured nutrients and biomass. And then finally, to also study the impact of treatments in the environment, I looked at the field study that I was telling you about out in the wetlands, and I measured the depth of litter layer in them. So the nutrients that I looked at are phosphorus and nitrogen. The phosphorus was diammonium phosphate and orthophosphate, which are plant available forms of the nutrient in soil and water. It can also exist in the world in a way that plants can't really use. So this is the way that they can use. And then I also measured total phosphorus to just look at everything in the environment. And then I looked at ammonium and nitrate for nitrogen, which are important nutrients for plants. I also collected soil samples to look at microbes. And so some of these are also important for microbes. We have that data. I have not analyzed it, so I won't really be talking about it. But I did measure things like pH. And then I also looked at soil organic matter, which is decomposing plant material once it starts to really incorporate into the soil. And then I also looked at litter depth, which out in the field, which again is just if you have a bunch of dead plant material before it becomes soil organic matter, it's a litter, it forms a layer of litter, and that can also impact nutrients through decomposition. It can impact soil moisture and microbes and things like that. So I looked at that as well. So my hypotheses for my first question. So looking at the control buckets, where I just let the plants grow, I predicted that over time, they would reduce the amount of nutrients in the soil because they're growing and there's no replacement. And then I also predicted that they would increase soil organic matter because bits of them die and start to decompose. So relative to that, the clipping and herbicide treatments I predicted would all increase or have more soil phosphorus at the end, because if the control methods control the plant, then the plant won't be absorbing as many nutrients. So there should be more at the end than if I just left the plants alone. Similarly, with nitrogen in the soil, the plants won't be taking up as much because they've been controlled. Similarly with phosphorus in the water and nitrogen in the water. Soil organic matter, I predicted would be less in the clipping treatment because the clipping removes some of what would otherwise die and incorporate in the soil. Whereas the herbicide kills the plant, produces a lot of dead plant material that will eventually become soil organic matter. And since the herbicide should kill the whole plant, but the clipping only removes some, I predicted that there would be slightly more soil organic matter in the combination treatment. And then litter out in the field where it was actually grazed and not clipped. Similarly, the cattle will eat dead stuff, the herbicide will kill it, and then the herbicide will kill more than the cattle eat. For my second question, I hypothesized that if West Indian marsh grass is better at taking up nutrients than maiden cane, which I thought it likely would, because maiden cane evolved in a low nutrient environment in Florida. And that environment is now becoming high nutrients, which means that other species that are used to higher nutrient environments are able to come in and invade because the native species don't really know what to do with all the excess nutrients in the environment. So if that's true, then West Indian marsh grass should have more phosphorus than maiden cane. And similarly, the nutrients in the buckets should all go into the plant. And so there should be less in the West Indian marsh grass buckets than in the maiden cane buckets. And finally, if West Indian marsh grass does outcompete maiden cane, then there should be more biomass of West Indian marsh grass. So to do all this, I first gathered soil from a wetland and mixed it all up and then put it in buckets. And mixing it up helped ensure that all the buckets were similar before I applied a treatment. So any differences I see later, I can attribute to what happened in between and not to the initial setup. I also collected initial soil samples and analyzed those. And that not only helps ensure, like it provides a baseline that I can compare to later, but it also helps really statistically ensure that, they are similar at the beginning. And spoiler alert, the buckets were all statistically similar before any treatment was applied. Then I collected plants from wetlands. This is West Indian marsh grass from a wetland on the ranch, and I planted them in the buckets. Here's the setup in the shade house, and then I let them grow for about a month, a little over a month, during which time I went into the field and helped Greg collect data on the NRCS Exotic Control Project. And then in July, I started to weed the buckets to make sure that only the two species I was looking at were present. Then I applied the herbicide. We use imazapyr out in the field. I used the same formula in the shade house. And part of that is blue dye so that you can see where it was sprayed and you can see in this picture, there's some blue on the buckets and the water is turning a weird color. And then I applied the clipping treatment. So I clipped with scissors flush with the top of the bucket, thinking that the cattle don't really shove their face deep into the water in order to eat. And then I took what I clipped and I dried it, weighed the biomass, measured nutrients in it. And then a month after that, so hopefully allowing enough time for any impacts to become measurable, I collected water samples, more soil samples, and analyzed them in the chem lab at the ranch. I performed statistical analyses. I looked at linear model. I looked at nutrients as a function of species, nutrients as a function of the two treatments, and nutrients as a function of species and the treatments. I ran one-way ANOVAs on the models. I tested that they were all normal, and I tested the interactions between the variables, which were never significant. So what did I find? The soil from the shade house, I found that herbicide treatments had more diammonium phosphate. So this graph is looking at just West Indian marsh grass and just the soil samples I collected at the end. So this is the final snapshot after all of the treatment effects have taken effect. And you can see that I have the lighter color is buckets that were herbicided, darker were not herbicided. The columns on the left were not clipped, columns on the right were clipped. And I found a moderate difference, moderately higher diammonium phosphate in the soil in the herbicided treatment. And then over time, this graph is looking at the difference between the final results and the initial samples. And you can see I have the soil control buckets in green, the purple is the maiden cane, and then the blue for West Indian marsh grass. And for the statistics, looking at the two species, I only looked at the clipped and unclipped. So those are the ones I could compare. And there was moderate evidence that West Indian marsh grass reduced it more than maiden cane did. Also about this graph, that line in the middle is 0, meaning there's no change over time. So if the error bars cross it, then they didn't change over time. You can see the West Indian marsh grass, clipped and unclipped, is in fact lower than that 0 line, whereas the maiden cane wasn't really. I also found that the herbicide treatments had more ammonium. I found pretty strong evidence, very strong evidence for this. You can see that the herbicide is much higher for the West Indian marsh grass. And then again, looking at over time and looking at the two different species, I found that West Indian marsh grass and maiden cane reduced ammonium similarly over time, which I think is interesting. So also the huge error bars on the soil are because a raccoon likely did its business in one of those buckets, such as the struggles of science in the real world. So you can take that with a grain of salt, but you can see that there wasn't really a difference between the maiden cane and the West Indian marsh grass. Finally, or not finally, I have a lot more results. The herbicide increased soil organic matter in West Indian marsh grass, but this is pretty weak evidence that this happened. And then looking at the two together, you can see that it was reduced in every single bucket, but that there's weak evidence for the maiden cane reducing it less than the West Indian marsh grass, which could be due to the fact that the maiden cane died back initially, and so there was more plant matter to become soil organic matter. So that you can also take with a grain of salt. Looking at the water results, I found that the West Indian marsh grass clipped treatments had more total phosphorus, which is interesting. This was, you can see that the columns on the right were taller, so there was more total phosphorus. I found that similar water total phosphorus, there were similar levels between the different species. So this is no longer change over time the way the other graphs were. This is now just mean water total phosphorus stocks, because I didn't have an initial. And then I looked at stocks, not concentrations, because there were differences in the water levels in each bucket. So that just helps take that into account. And there's weak evidence that the clipped total phosphorus was less than the soil. But interestingly, there was not that similar impact on the maiden cane of clipping or not. I also found that the herbicide treatment had more ammonium for the West Indian marsh grass. And there's very strong evidence for this, as you can see here. So similar to the soil, there was also very strong evidence for that. But then looking at both species, again, you can see that herbicide is much taller, and you can see that the soil control bucket is also much higher because of that raccoon. And there's very strong evidence that both species had less ammonium than the soil, and weak evidence that maiden cane had less than West Indian marsh grass. Looking at the field, I found that grazing reduced litter and herbicide increased litter. There was very strong evidence that the grazing reduced the litter and strong evidence that the herbicide increased the litter, as you can see in this graph. And then also looking at this picture, you can see what the litter layer looks like out in the field versus the living plants. And this is one plot in the project. Looking at the biomass or the clipped plant results, I found that West Indian marsh grass produced more biomass than maiden cane. There's strong evidence for that. And this will likely become stronger over time because my results don't show a whole growing season. And West Indian marsh grass grows taller than maiden cane, so this will likely become an even stronger effect. And then interestingly, I found that West Indian marsh grass had lower total phosphorus than maiden cane. So this is looking at stalks, which again takes into account the difference in biomass. And so maiden cane has so much more phosphorus in it that even though there's less biomass, it would still contribute more than West Indian marsh grass, which is really interesting. And there's moderate evidence for this. But what does this all mean? So returning to my hypotheses, which are in the turquoise, the yellow is what I actually found. Again, this is all relative to doing nothing and just letting them grow. I found that clipping didn't really have an impact on phosphorus in the soil, whereas herbicide did. Same thing with nitrogen in the soil. Clipping didn't really have an impact, but herbicide did. Interestingly, for phosphorus in the water, herbicide didn't really have an impact, but clipping did. Nitrogen in the water, clipping didn't really have an impact. Herbicide had an impact. Soil organic matter, clipping there wasn't much of a difference between doing nothing and the treatment. Whereas herbicide, there was more soil organic matter. And then litter, clipping, there was less. Herbicide, there was more. And interestingly, the combination effect, the grazing actually overpowered the herbicide. And so there was less litter there. Looking at my second question, I thought West Indian marsh grass would have more phosphorus. It did not. Maiden cane did. And then looking at the nutrients in the buckets, that was more of a mixed bag. Diammonium phosphate and soil organic matter, there was more of that in the maiden cane buckets, but there was less ammonium in the water in the maiden cane buckets. And then the rest were the same. Nitrate was actually 0 in all of the buckets that I measured, which was interesting. And then finally, I found that there was more West Indian marsh grass biomass. So sort of looking, what can we conclude from all of this? The treatments do have an impact. Specifically, the imazapyr, the herbicide, has nitrogen in it. So from these results, I can conclude that it actually adds nitrogen to the environment, which is important to consider. It also stops nutrient uptake, which is likely why the other nutrients were higher in the herbicided treatments than in the non-herbicided. Whereas again, with the nitrogen, it was actually added instead of just stopped being uptaken. The herbicide also increases litter and the grazing reduces litter. I also found similar uptake abilities between West Indian marsh grass, the invasive, and its native equivalent. However, there wasn't quite enough time to see all of the effects that there could have been. Growth stage matters for nutrients and forage. So potentially, if I had harvested the plant material at a different time of year, I might have found different results. I also have not yet analyzed the microbe data, which could help explain potentially some of the soil organic matter results and why the nitrate was all 0. So denitrification is when microbes eat soil organic matter and convert nitrate into nitrogen gas, which then escapes. And this happens in waterlogged soils, which is what my buckets were. But without that microbe data, that's still just speculative. I finally also used soil at the beginning that was low in nutrients in order to try to measure an effect, but potentially they were all used up. And so maybe that's why there wasn't a huge effect, or I didn't see much of an effect from some of the treatments. So next steps for this, I'm very excited. Greg is continuing the project and will collect data again around the next, or the first big frost of the season. And hopefully that will be enough time to see more of an effect. He'll also include the whole plant in order to get a phosphorus budget, which will kill the plants and the experiment, which is why I didn't do it. It would also be interesting if we could also look at nitrogen in the plants. And then we'll also hopefully apply these findings to the field and calculate how much is removed from grazing, how much stays because of herbicide or is added because of herbicide. Of course, the other side to the cattle grazing is that the cattle also poop. And so how much nutrients are returned by the cattle is another really, I think, important question to answer. And then also figure out if these amounts are enough to seriously impact nutrient levels and impact management. And finally, we'll look at the microbe data, and it would be good to look at the interactions between the microbes, nutrients, and the treatments. Thank you everyone for coming to my talk 2 months later. Especially thank you to everyone at Buck Island Ranch, from operations to the lab, Betsy and Greg for advising me, and Carly for being a great sugarcane boss, Mandy and Hayu for helping me out in the chem lab, Mardia for helping me brainstorm, and then Akeem, Lydia, Alma, Chef, Kyle, Claire, and Emily for helping do boring things like label specimen cups and whatnot. And thank you to Cabby and the Sugarcane Project for funding me. And yeah, these are my sources. Thank you. Does everyone have any questions?
So Luca, we have about 11 people in here, just so you know, in our watch party. So,
There is one question.
yeah, I'm going to work on getting the camera and the microphone set up for them to ask questions, and Greg, you can do yours.
All right. So it's a question from Akeem. Do you have any idea what factors allow maiden cake to take up more phosphorus than West Indian marsh grass in your shade house experiment?
I do not know. I guess one thing that we were thinking is that potentially it could just be different stages of growth for the different plants, where maybe when the plants that I harvested from the field and then planted, whatever growth stage they were in might have just been they might have just taken up more nutrients at that point. But if we look at it over the whole, it's like a whole growing season, a whole year, we might find different results. So I don't know.
I guess I can ask another. Oh, Laura, we can't hear you.
No, I was just telling them they have to really project to hear.
Right. Do we have a question from the audience from where you are, Laura?
Yeah.
All right. So you said that maiden cane took up more phosphorus. But did you say that in the soil and in the water, their phosphorus levels were about equal? That's what I thought I saw, but maybe you moved it too quickly for me to see. So if that's so, where is phosphorus? Like, what explains the difference? Or rather, why would their phosphorus level be the same if maiden cane is picking up more?
Yeah, I have no idea. I think it's, yeah, part of it is that some of the nutrient analyses, there could have just been some errors in those. And then also, Yeah, I'm not, oops, not 100%. I don't remember about the soil levels. But part of it could have gone into water, and we might have lost some of the water from some of the rain and things like that. There was also, oh yeah, it could have also just depleted it equally. I don't know. There could have been a lot of different explanations.
Go ahead, loud.
So to piggyback off of Jeffrey's question, what part of the plant tissue did you analyze for phosphorus? Is it the leaf, just the leaf, or did you also analyze the roots? Also, just a comment, I really like the way that you just graphically displayed your question and hypothesis. It was really easy to follow, and so good job with that.
Thank you, I appreciate that. I looked at the forage, which is like plant and stem, or like leaf and stem. I did not look at the roots. That's one of the things that Greg will look at differently. But yeah, I didn't want to harvest the whole plant because that would have killed it and ended the experiment.
Right.
Anybody else? All right, I think we don't have any other questions here.
All right, it seems like online it's the same thing. I just have one question, I guess, since usually there is another one. Another question from my team. Why do cows favor West Indian marsh grass more than maiden cane if maiden cane is more nutritious? Is maiden cane more nutritious actually, or is it only phosphorus? It's a trick question. Is West Indian marsh grass still more palatable?
I guess so. West Indian marsh grass has the triple effect, I guess, of being palatable, digestible, and nutritious. So maiden cane might not taste as good, or might be harder to digest, even if there's more nutrients in it. And then, yeah, I guess to answer a few questions ago, there could have been a difference in that maybe West Indian marsh grass had more nutrients in the roots that we didn't measure. And so that could help explain the difference. But yeah, I didn't measure palatability or digestibility. So yeah, I don't know. That's what the literature says about the different plants.
I have another question from Christine. What was your favorite part of your project?
I don't know. I think it was all pretty cool. I liked going into the field and collecting this stuff, the soil and the plants, and then growing it. This project was good for my professional development and learning that I don't love doing chemistry analyses. So that was, being in the chem lab a lot was not my favorite part. But it was really cool to learn all the methods and stuff. But yeah, I guess I like being in the field more.
All right. I think that's it for the question. The only one I could ask is, what was your favorite plant? Usually that's Mandy that asks that question, so she's not here.
We do have one more over here.
I was curious how your results may play into people arguing whether they should be treating West Indian marsh grass or not. Versus, whether if it's fine for the cattle, if people are more inclined to leave it, or like, how do you think your results would fit into that broader argument?
Yeah, I mean, so that was definitely something that I was hoping to be able to do, is like provide management recommendations. I don't think the results are particularly conclusive. Hopefully, with this final round of sampling, Greg might find something more. And then also the microbes might also reveal something else. But I guess the thinking was that if West Indian marsh grass takes up more nutrients from the environment than maiden cane does, then that's another benefit. Not only is it good for the cattle, but it can also like help clean the environment. Since that's not what I found, I guess that's more evidence to help control it. But again, there's still a lot more to be done with the data, and hopefully with more time as the experiment's gone on, the results can be a little more conclusive and help come up with concrete management recommendations.
All right.
Okay.
Thank you, Luca.
Thank you. I don't know what my favorite plan is. The maiden cane was really pretty. The West Indian Marsh Grass was fun to feed to the cattle. Yeah, I like aspens.
All right. Well, thank you again, Luca. Thanks everybody for attending here and online.